Hello everyone, this is Josephat Kimani. Today we'll be going through the histology revision series. And today's scope of the session will cover on epithelium, that is lining epithelium, glandular epithelium. We'll revise on connective tissue, and then we'll have some few questions covering the scope of the sessions today. So we'll start us off by checking the various types of epithelium. At this point, you can appreciate this is epithelial tissue, which lines the various tissues and cavities. So this is simple squamous, simple cuboidal, simple columnar, stratified squamous, stratified cuboidal, stratified columnar, pseudostratified columnar, and transitional epithelium. So a point to appreciate is that this simple squamous epithelium, they will be found in the vascular system. So this simple squamous epithelium will be found in the vascular system, that is the endothelium, be found in the body cavities. Remember, body cavities are lined by mesothelium. Then you'll find it also in the Bowman's capsule, in the kidneys, and then also in the alveoli, in the respiratory spaces of the lungs. And the major functions of this simple squamous epithelium is to enable the exchange and lubrication. So these two, the simple squamous allows the exchange and lubrication of tissues. For simple cuboidal, this simple cuboidal will be found in ducts of small exocrine glands. They will also be found in the kidney tubules, the renal tubules, and also in the thyroid follicles. So that's where you'll find the location of the simple cuboidal. Function of it is that allows absorption and secretion of it. Then you have simple columnar. For simple columnar now, this one will be found in the GIT majorly. So talking about the small intestine, the colon, the stomach itself, the large intestine itself, it will have the simple columnar, and also you'll have even the gallbladder having the simple columnar epithelium. So the function of this simple columnar epithelium is to allow absorption in the GIT, secretion in the GIT, and also both of them. So you can have either absorption in the stomach, secretion in the gallbladder, and also both of them absorption and secretion in the large intestine. So stratified squamous epithelium, very important. You can have stratified squamous keratinized, stratified squamous and keratinized epithelium. Stratified squamous epithelium, the one that is keratinized, we found in the epidermis. And then the one that is non-keratinized, we find it in the oral cavity and the esophagus. You can also find it in the vagina and also in the ectocervix itself. That is stratified squamous non-keratinized epithelium. Keratinized epithelium is only found in the epidermis of the skin. For the stratified cuboidal, this one will be found now in the sweat glands, usually. They will found it in the sweat glands, and also you find it in the large ducts of exocrine glands. Remember, for simple cuboidal, you find it in small ducts of exocrine glands. For stratified cuboidal, you find it in large ducts of exocrine glands. Another function of stratified cuboidal acts as a barrier and also for protection. We can go all that and talk about pseudostratified columnar, which is very important. So remember, pseudostratified columnar, you can either have it as pseudostratified columnar ciliated epithelia with goblet cells, a characteristic of respiratory epithelium. And then if this pseudostratified columnar epithelium doesn't have goblet cells, you'll find it in olfactory epithelium. And that's the reason why the location of this pseudostratified columnar epithelium will be located in the trachea and the bronchial tree, and also in the olfactory region. Point to appreciate is that even the epididymis, sometimes the different tubules of epididymis will have a pseudostratified column. Transitional epithelium, a characteristic of the urothelium, will be found in the urinary system. So talk about the urethra, the ureter, and the bladder itself. So we can talk about apical modifications of the epithelium. So this is the apical modification. Remember, apical modification can either be 3. sorry, they can either be four. You can have keratin, you can have microvilli, you can have cilia, and also you can have stereocilia. For lateral modifications, you have junctions, the junction of various junctional complexes. You can start with a tight junction, which prevents the transcellular movement communication, just allows the paracellular movement of substances. And then you have adherence junctions, which joins various neighboring cells. That's the reason why they're called adhering junctions, because they adhere to one another. Then you have desmosomes. So desmosomes joins the intermediate filaments in one cell to another, to a neighboring cell. And for the gap junction, at this point, you can point this gap junction. They allow transmission of solutes and substances, water-soluble ions and molecules from one neighboring cell to another. So the last modification, you have the hemidesmosomes and focal adhesions. So the hemidesmosomes themselves are found on the basement wall. So this one will anchor the intermediate filaments in a cell to the basal lamina. So remember the basal lamina, the basal membrane has various parts, the lamina rara and the lamina densa. Yeah, so, and also the basal lamina itself. So that's a very important component of, of the basal of the basal modification. Then these are better image showing in the electron microscopy of the same. These are tight junctions. You can see it there. And these are the adherent junctions. And then these are the desmosomes. You can see the classification and the various epithelial specialization. It's noted there. So we have cilia, microvilli, stereocilia, and keratin. For the lateral domain, we have zonular occludence, zonular adherence, macular adherence, and the gap junctions. And then for the basal domain, we have hemidesmosomes, focal adhesions, and then basement membrane ideally holds the epithelium uh, via now these hemidesmosomes and the focal adhesions. For glandular epithelium, I would like us to divide it into endocrine glands and exocrine glands, with the major difference being that endocrine glands, they secrete their secretions in blood. Exocrine glands, they secrete their secretions using ducts to their target uh, structures. So exocrine glands can either be unicellular or multicellular. Based on the structure, you can have simple glands or compound glands. Based on the secretory unit, you can either have tubular, alveolar, or maybe mixed tubular alveolar. So remember, alveolar is also known as a sinus. Based on their mode of secretion, you can either have merocrine, holocrine, or apocrine. A better illustration of the cell. So on the proximal aspect, you can appreciate these as simple glands. Based on the ductal system, you can appreciate the ductal system is not branched. So if the ductal system is branched, we'll have the compound glands at this point. Another point to appreciate is that for the secretory portion, it can either be tubular, like in this simple tubular, can either be coiled, simple coiled, but tubular morphology. Then if the cells are somewhat triangular in shape, you'll have simple acini. So simple acini, you'll appreciate the triangular shaped cells. Then if you have mixed, you'll have compound or maybe simple tubular acina. So for example, this one is compound tubular acina. So at this point, you can appreciate a good example of compound but acina, compound but a tubular gland, or compound but a tubular gland. You can appreciate we have the mucus glands majorly in the mouth and the bulbourethral glands in the male reproductive system. But the compound acina or the compound alveolar can have mammary glands. For the compound tubular alveolar, you'll have now the salivary glands and also the pancreatic cells. For simple glands, simple tubular, a good example of is the intestinal glands. simple assigner. A good example of a simple assigner is that you find it in simple branched glands in the body. Yeah, and then for the simple coiled tubular glands, you'll find them in the merocrine sweat glands, as you saw earlier. For connective tissue, its revision to appreciate is that connective tissue is made up of cells and extracellular matrix. The extracellular matrix will have fibers, will have multi-adhesive proteins, and then it will also have some proteoglycans. So the cells that you might have, you can have adipocytes or the fat cells. You can have mesenchymal cells. These are stem cells that give rise to all the other cell types. You can have fibroblasts. You can also have mast cells. You can have mast cells. You can have other cell types that know regarding which connective tissue you're talking about. And then for the fibers, you can have reticular fibers. You can have uh, elastic fibers as shown there, and also you can have collagen fiber. So through now the various percentages of these will determine which connective tissue will have. So as shown there. So let's delve to some few questions as we try to finish up the session. Is that the question is identify the type of connective tissue, give reasons and location. So this is dense regular connective tissue, a characteristic of tendon, aponeurosis, and ligament. So those are the locations. You can appreciate it's fibrous, and then there is like parallelism of the fibers as shown there. Let's say the epithelium shown, give reasons and location of the epithelium. At this point, I'd like us to appreciate you have cells originating from the basement membrane. So this is the basement membrane. Then you can appreciate a brush border. Then you can appreciate you have some kind of goblet cells at this point. And then a point to appreciate is that on the uppermost part, we don't have nucleus. So at this point, this is pseudostratified columnar epithelium ciliated with goblet cells. You can also appreciate it is hyaline cartilage, just trying to show us that this is the respiratory epithelium. Identify the type of connective tissue, give reasons and locations. At this point, you can appreciate this mesenchymal connective tissue, a characteristic of in embryo, it's found on the umbilical cord, but in adults, we find it in the nucleus pulposus, in the intervertebral disc, and also in the tooth pulp of the tooth. Identify the epithelium shown, give reasons and location of the epithelium. The epithelium shown at this point, I like us to appreciate, first of all, is stratified. Then you can have bumpy appearances on the apical end. You can appreciate binucleation. So these cells that are here just tries to show us like one cell, the apical cell is binucleated. This is one cell that is binucleated. This other cell is binucleated, and this other cell is binucleated. A characteristic of transitional epithelium or the urothelium that is present in the urinary system, the bladder, the ureter, and the urethra, the proximal urethra. At this point, this is another epithelium that I'd like us to appreciate. So this is keratin. You can appreciate the stratification that is there. And then you can appreciate on the apical portion, the nucleus are squamous in nature. So you can appreciate the nuclei of the apical cells are squamous in nature. So this is the epidermis. The epidermis has 5 layers. Then you'll have the dermis. This is the dermal papillae. And then these are the epidermal projections into the dermis. So at this point, we'll have a certain receptor known as Mesner's receptor. So at this point, this is stratified squamous keratinized epithelium, a characteristic of the epidermis. Identify the epithelium shown, give reasons and location of the epithelium shown. So at this point, I would like us to appreciate that we had seen this earlier. But a point to appreciate is that this is pseudostratified columnar epithelium. You can appreciate a brush border. This is cilia. And then you can appreciate that there is no goblet cells. Remember, for pseudostratified columnar epithelium is either found in olfactory epithelium or in respiratory epithelium. So we can appreciate presence of Bowman's glands, ducts of Bowman's glands, you can appreciate them there. And then you can appreciate some olfactory nerves at this point. So this olfactory epithelium, characteristic of the superior portion of the nasal cavity. This is simple cuboidal epithelium. So we say for simple cuboidal epithelium, it's located in the small ducts of exocrine glands, also located in the kidney tubules, the renal tubules, the thyroid follicles, and also in the germinal epithelium. So surface of the ovary, the germinal epithelium will have simple cuboidal epithelium. So at this point, I would like us to appreciate the nature of this. So identify the epithelium shown and give reasons and location of the epithelium. So if any one of us can give the correct structure of what this is a cornea. So for the cornea, you can appreciate it has an epithelium and then an endothelium. So this endothelium is lined by simple squamous epithelium. Then it will have a Descemet's membrane, then it will have substantial structure with collagen fibers, and it will have a basement membrane, and then it will also have the epithelium, which is stratified squamous, non-keratinized epithelium. So this is characteristic of the cornea of stratified squamous non-keratinized epithelium. It's one of the epithelium on the outside. The epithelium on the outside, that is on the inside, that is endothelium, will be a characteristic of simple squamous epithelium. And then we talked about the other layers of the cornea. So this similar case as the one that we covered earlier, you can appreciate that this is stratified squamous non-keratinized epithelium. So zero modification, we can't appreciate keratin. So we say that stratified squamous non-keratinized epithelium will be found in the ectocervix, in the vagina. We also say that you find it in the masticatory epithelium of the oral cavity. You'll also find it in the esophagus. So, and also in the anus, that is the location of stratified squamous non-keratinized epithelium. So those are the locations of the stratified squamous non-keratinized epithelium. So these are similar one as the earlier one. You can appreciate all the cells are originating from the basement membrane. You can appreciate presence of goblet cells. You can appreciate a brush border, a characteristic of pseudostratified columnar, ciliated epithelium with goblet cells, a characteristic of respiratory epithelium. At this point, you can appreciate this. You can appreciate this is pseudostratified columnar epithelium with stereocilia, a characteristic of the epididymis. Pseudostratified columnar epithelium with stereocilia, a characteristic of the epididymis itself. You can appreciate the stereocilia at this point. Similar case, pseudostratified columnar epithelium, ciliated, zero goblet cells. You can appreciate the apical points, has no nuclei. Then you can appreciate the presence of presence of a brush border, the cilia. And then at this point, you can appreciate a very large nervous bundle. So this is a olfactory nerve. Then you can appreciate the Bowman's glands, a characteristic of the olfactory epithelium. So this epithelium is referred to as a pseudostratified columnar epithelium, ciliated without goblet cells, a characteristic of olfactory epithelium. I presume at this point you can easily appreciate what is this. You can appreciate this is keratin, or this is apical modification. Then for A, that is the epidermis. You can appreciate this dermis, and then is the dermal papillae, these are epidermal projections. And then when we look at this, ideally this is the Meissner's corpuscle that we are talking about, a receptor. Identify the type of connective tissue given and give reasons and locations. So ideally at this point, This is skeletal muscle. Skeletal muscle is very much important because it can easily be confused with dense regular connective tissue. So appreciate the striations, appreciate the peripheral nucleation, appreciate its multinucleated, appreciate the fibers are parallel to one another, and then also appreciate that there is no branching of these fibers. At this point, I'd like us to appreciate what is this. So this is dense, regular, connective tissue, a characteristic of tendons, ligaments, and aponeurosis. So I had seen this earlier, so I won't delve much into it. Identify the type of connective tissue. Ideally, these fibers. These are reticular fibers, which are found in the reticular endothelial system. So talk about the liver, the spleen, the lymph nodes, the lymphatic vessels. All of them will have the reticular fibers. At this point, you can appreciate this is a portal vein. Sorry, this is a central vein. The central vein, and then on this other end, you can appreciate the portal triad. So the portal triad has a branch of the bile duct, has a branch of the portal vein, and then has a branch of hepatic artery. So that is a portal triad. And then this is the central vein, and then these are the hepatocytes, the cells of the liver. So this organ ideally is the liver with the reticular fibers. These are repetition of the same pseudostratified columnar epithelium, ciliated without goblet cells, are characteristic of olfactory epithelia. At this point, I'd like us to appreciate this. This is still the same, but now this one has goblet cells. So this one is pseudostratified columnar ciliated epithelium with goblet cells. Various types of glands. You can appreciate this is simple tubular gland, simple acinar gland, and then this is simple tubulo-acinar gland. You can appreciate there. So the various types of glands. But now this one is simple but branched. Simple but branched type of gland. So this one is not compound, it's simple. Then for the electron microscopy, you can appreciate that this is the mitochondria. And then these are the rough endoplasmic reticulum. Remember, the function of mitochondria is that apoptosis happens there. So it's the one that provides the stimulus for apoptosis. Then you can also have culture storage. Oxidative phosphorylation happens there, and then it also contains genetic material. That is the functions of the mitochondria. For the rough endoplasmic reticulum, a point to appreciate is that this rough endoplasmic reticulum participate in protein synthesis of proteins that will be packaged and to be used extracellularly. So that's the function of rough endoplasmic reticulum. Can appreciate is still the same. This is an electronic microscopy image of the nucleus. You can appreciate the nuclear membrane, and you can appreciate the nucleolus. Then B is the nucleoplasm, and then A is the chromatin material. So those are the functions. Those are the structures that can be easily identified in an electron microscopy regarding the nucleus. At this point, I'd like us to appreciate the apical modifications of the epithelium. So this point is a microvilli for absorption, and then this is stereocilia. So A is a microvilli and B is stereocilia. Of course, at this point, I'll urge us to go through the various locations of microvilli and the various location of stereocilia. So, but a point of appreciation is that microvilli will be found majorly in GIT and even in the urinary system, the kidney, the proximal convoluted tubule, and the distal convoluted tubule. And the stereocilia will find it in the epididymis. The same connective tissue, the fibers at this point, like us to appreciate these are the reticular fibers, which are found in the reticular endothelial system. They will also be found in other organs such as the spleen, the lymph nodes, and the lymphatic vessels. Also, a point to appreciate is that some fibers Some arteries might have the same structures. This is an electron microscopy of seriocilia, as shown there on in the epidermis. Characteristic we can easily appreciate. Then, almost finishing up, this is rough endoplasmic reticulum. Of course, we talked about it. Packaging involving protein synthesis of proteins that will be used extracellularly. Then free ribosomes will be used for the synthesis of proteins that will be used intracellularly. So free ribosomes, proteins that will be used intracellularly. Rough endoplasmic reticulum, protein synthesis that will be used extracellularly. Then at this point, we can wrap it up with identifying this. This is a transverse cross-section of cilia. So the 9 plus 0 plus 1 rule, you can appreciate it here, an electron microscopy of the cilia. So those are the answers provided. Those are the answers for the questions provided. You can always go through them at your point, and then all the best in your studies and have good luck and looking forward to the next session that you're going to have. All the best in your academics.